research as a soil microbiologist working in agriculture is really focused on trying to understand how soil microorganisms are working within a soil environment and with plants, and that if we can understand these relationships a little bit better, then maybe we can manipulate things and use that information to improve crop production. Can we make this, can we produce crops in more sustainable or better improved ways by understanding how those soil microbes are interacting with the crop itself? We're looking at a variety of different ways to do this. One of the big things that we're focusing on is with cover crops, and so cover crops are a relatively common technique in the Midwest and with vegetable and row crops as a way to improve soil health and soil conditions. But they haven't really been looked at much in South Florida, and they have not been looked at much for citrus production, and so, or tree crop production in general. And so we've been really focused on trying to both figure out and optimize how do you grow cover crops in citrus, and also what are the changes that they are making and how are they impacting that soil environment. One of the other big things that we've been working on is with biological soil crusts. These are, or biocrusts, these are communities of bacteria, fungi, lichens that grow on the top of soils and sort of form this consortia of organisms. They're really common in desert and arid ecosystems around the world, and that's where they've been most studied. And one of the big things in those ecosystems is that they are major sources of nitrogen for those ecosystems. And so when I started here, a grower actually came to me and said, "Oh great, you're a soil microbiologist, there's something I want to show you on the ground." And I said, "Well, what on earth am I going to be looking at with on the ground for microbiology?" And that I could see with my naked eye. And sure enough, there were what looked like these biological soil crusts in a citrus grove. And so we started looking at that and got a grant to look at that, and it's really been one of the first studies to explore and try and understand why and how these biological soil crusts are growing in agroecosystems, because this is an environment that has a lot of nitrogen and water inputted into it, unlike these arid deserts. So why are there still these nitrogen-fixing organisms growing right underneath the trees? And so we've been trying to figure out what's going on, and have had 2 students working on that project. One of the other newer project areas that we're working on is trying to understand soil fumigation impacts on microbes. And so fumigation is a technique that's been used for 50 plus years to control different pathogens and weeds, nematodes, things like that in vegetable production, where different chemical compounds are basically injected in the soil to try and kill those organisms and control those organisms. But strangely, despite the fact that this has been done for 50 plus years, there's not a lot of information on what's happening to all the other organisms in the soil. And so we've got a new project trying to figure that out. And so if we can understand what changes are happening to the soil microorganisms when we apply these compounds, maybe there are things that we could change a little bit and make this more efficient or more sustainable. But I think you have to understand what is happening first before you can start manipulating things. In microbiology and microbial ecology, we're still exploring a lot of unknowns, but it's also, I feel like, at this great boundary between basic and applied research, and that we know if we could answer some of these basic questions, we can use that as applied to make applied changes. And so I think most of my projects, I think you could qualify and categorize as being at that edge between basic and applied, and that if we want to make changes to how a particular practice is occurring, we need to understand what is happening first. And so some of that is basic microbiology knowledge, and that we just don't understand some of these interactions yet. But hopefully once we do, then we can apply that knowledge. And so I find it really exciting exciting time to be in microbiology and microbial ecology. A lot of the techniques that we're using are things that have really just become more common, particularly in soils in the last 5-10 years. And that these DNA-based techniques, some of them weren't around, or at least weren't affordable when I was in grad school. And now they're becoming more affordable so that we can really use them and start to figure out these details that we really just couldn't look at previously. We now know that there is a lot of stuff we don't know. We've now been able to find out that, most of the soil microorganisms you can't grow in a lab. So we have to use these DNA techniques to try and figure out what's going on, and realizing all the time that things are more complicated than we thought. In addition to graduate students, we also have a great program here for undergraduates, even though we are far away from Gainesville. Ave Maria University is located just down the street. It's about 20 minutes away, and they don't have a graduate program at all. And so we've got a program with their biology department where students there can earn credit, research credit, by working in some of the labs here at the center. And so I currently have one student who is doing that. And then I also have a student who is working on his associate's degree at FSW, and is working in the lab as well part-time. And so there are definitely opportunities for undergraduates. We certainly would love folks to come down from Gainesville too, and there are summer programs available for that as well.